So my topic was on developing novel HER2 inhibitors to overcome drug resistance of current therapeutic drugs to tucatinib with a double mutant L755H, D769H using virtual screening and kinase inhibition assay. So a little bit about HER2. It is the human epidermal growth factor receptor, and it is part of the tyrosine kinase receptor family. And HER2 is essential for cell proliferation, differentiation, and survival. It basically controls how healthy breast cells grow, divide, and repair itself. However, there are mutations that occur in HER2, and they account for about 10 to 20% of breast cancers. In this case, we are specifically looking at mutations L755S and D769H, and it's basically changing leucine at position 755 into serine, and then changing aspartate at position 769 into histidine. So methods we used were database searching, protein structural properties, homology modeling, docking, and virtual screening. And from the results, we would try and find ligand interaction diagrams, and you can see that there's a ligand interaction diagram for wild type HER2, and you can also see that 2 ligands are mutated, and the mutated ligand that was specifically for L755S was about -10.587, and the mutated ligand for D769H was -9.338. And these are the zinc compounds that were used from the bioinformatics student. And he also put the percent oral bioavailability, and that's another reason that you can see that a zinc compound is helpful and it's actually working, because if you have a higher percentage of oral bioavailability, that means that it's less amount of the drug needed to actually treat what's going on. And it is usually, if you have a higher bioavailability, it means that there are less side effects associated with it. And with the expected results, we would expect better IC50 values with the zinc compounds than with the drug or the mutants, because we want to see that it actually is inhibiting correctly, and it takes less concentration to do that. And this also shows like the kinase assay of how we would, the pattern we would want to see, and we expect to see with the table, we kind of want to see the same type of numbers going on between the mutant and the wild type, and we would want to see better results from the 5 compounds. And these would be examples of IC50 graphs we would like to see if we were using zinc compounds that were found before. That's my references.
No, I mean, the ask question. Your turn?
I don't have any questions. It was really good.
Really good, that question. Okay, so my kind of the comments is that put unit to those docking score, typically the IC50, docking score kilocalorie per mole, and IC50 typically is micromolar or nanomolar. Put units with those numbers.
Okay.
Yeah, that's it.
Perfect. Thank you.
Okay.